left and left to right.
He's coming from warm Cleveland. Now, I moved here because I ran away from the snow. Now he's telling me this is colder than Cleveland today. But anyway, Ken and I go back many, many years. We were together at Case Western Reserve in Biomedical Engineering. And Ken is a faculty member now in Biomedical Engineering and Cardiology, Medicine. And also this Heart and Vascular Center at the MetroHealth Medical Center in Cleveland that is headed by David Rosenbaum, who we had several weeks back. Ken has done a lot of work in cardiac electrophysiology, and in recent years, his emphasis was on calcium imaging and arrhythmias in the context of calcium cycling. And so he'll tell us today about calcium-mediated arrhythmogenesis and myocardial infarction. Thanks for coming.
Thank you, Yoram. Also, thank you to everyone for spending time with me today. I know you're very busy, you have a lot of things on your mind. I appreciate the time you spent with me. Everyone has been great. The interaction I've had has been really wonderful, and I've been very pleasantly surprised. You all should be very proud of the work you're doing and proud of your center. It's wonderful. So thank you again. So I'm going to be talking about calcium-mediated arrhythmogenesis and myocardial infarction. And a lot of the work that I'm going to show you today is fairly new for me. Some of it will be based on previous work we've published. Some of it will be work that we've published in abstract form, and then some of it will be unpublished. So it's very exciting for me to present a lot of these data to you, and I welcome your feedback. So if I asked a physician, maybe none of the physicians here, I heard there are clinical people in the audience, so of course no one here. If I asked you, give me an example of a calcium-mediated arrhythmia, I think the first answer I get would be, well, it's either associated with digitalis intoxication or now CPVT. That's one that they remember. But I'm not going to be talking about either one of those. Heart failure, maybe. It's associated with mechanical dysfunction. We know because changes in calcium regulatory proteins. Tachycardia-induced, it's not chronic MI, so I'm not going to be talking about that either. I'm also not going to be talking about acute ischemia. Acute ischemia is associated with a lot of changes in calcium regulation. Again, what I'm going to be focusing on is chronic MI. There is not much we know about calcium-mediated arrhythmogenesis in chronic MI, and it's surprising because chronic MI is the condition that kills most people. So that's what I'm going to be focusing on. And the model that we use is a rat model of a permanent LAD occlusion. You can see here that after about 2 to 4 weeks, you get some significant thinning of the wall. If you wait much longer, the wall gets very thin. It's almost like a 1 millimeter thin. I was actually pleasantly surprised when I was here. I didn't think anybody would really be talking to me about scars and infarcts, but it appears as though there's a lot of people interested in this. And you can see the scar from the epicardial surface. So it's fairly visible. And there's a shortening fraction associated as well with this model. So now this is typically what you would expect with a model of MI. Shown here is the same picture as I had before. Here's an optical mapping field. And these are isochrone maps of activation during pacing with a few representative action potentials shown on the side. You can see conduction slowing near the infarct border zone. And as I've spoken to several people here today already, you do measure action potentials from m-SCR, albeit they're much smaller action potentials. And if you apply a premature stimulus, 1 or 2 premature stimuli, you can get functional reentry. So this is very typical of this model. You can get this. This is a very important substrate for arrhythmias associated with MI, but this is not what I'm going to be focusing on. What I'm interested in is calcium-mediated arrhythmias. So if you were to open up a medical textbook, this would be the mechanisms of arrhythmias. You have abnormalities of impulse formation, impulse conduction, and also abnormalities of impulse formation. So where does calcium-mediated arrhythmogenesis fit into here? Well, most likely, most people would say, oh, delayed after depolarizations and spontaneous calcium release, that's for sure. So that's one of the substrates that I'm going to be talking about. The other one may not be as obvious. There is a calcium-mediated substrate associated with reentrant excitation, and that's cardiac alternans. So what's really nice about thinking this way, if you're very interested in calcium and its role it plays in arrhythmogenesis, you can attribute it to not only abnormalities of impulse formation, but also reentrant excitation as well. I'm going to just very briefly talk about the substrates. You may have heard this already, you probably know about it already, but for those who don't, cardiac alternans is most often described on the ECG as T-wave alternans. You have an alternation in the T-wave amplitude, which leads to sudden cardiac death, can lead to sudden cardiac death. It's also used as a measure to predict risk for sudden cardiac death. Both our laboratory and other laboratories have shown that the alternations in the T-wave morphology are due to alternations in action potential duration, short action potential, long action potential, short action potential duration. And during action potential duration alternans, you can get very large gradients of repolarization, which has been shown to be sufficient to cause sudden cardiac death. And what causes action potentials to alternate? Well, our laboratory has shown that calcium transient alternans is one mechanism by which you can explain action potential duration alternans. Smaller calcium release, shorter action potential duration, larger calcium release, longer action potential duration. This is not the only mechanism of alternans, but it's one mechanism of alternans that we think is important, especially in disease. And as I mentioned earlier, this is an important mechanism by which abnormal calcium handling can cause reentrance excitation, something that people don't often connect. Yeah.
So the question is, the calcium trans and normal is derived from the action potential prolongation, or which one is the original?
Yeah, that's which comes first.
Yes.
Yeah, that's a question that a lot of people are still investigating. I don't think there's any experiment that has explained this definitively, but a majority of the evidence suggests that calcium transient alternans is occurring first, and that's causing action potential duration alternans. But there's ways in which you can explain the opposite as well, although it can be both. Here's the other substrate, delayed after depolarizations. This is a classic example of an arrhythmia initiated by delayed after depolarizations. You pace rapidly, you stop pacing, 2 extra ectopic beats, and then you get these really wonderful oscillations of membrane potential. These are the delayed after depolarizations. And in most cases, we think that this is due to spontaneous calcium release, which I'm going to abbreviate as SCR. You'll see that a lot in my slides. Now, the spontaneous calcium release that we measure is with optical mapping techniques. We don't use microelectrodes. We don't measure fluorescence of calcium from an individual cell. We measure calcium in the whole heart, and one recording one recording site, one pixel, is recording calcium from thousands of cells. Okay? So what we're actually measuring when we measure spontaneous calcium release is calcium from multiple cells. So what I like to do is call this an m-SCR, just to differentiate this from just normal spontaneous calcium release. And in one study that we recently published, we showed that this has some special properties, but I'm not going to be talking about this. As far as this talk goes, these spontaneous calcium releases in tissue behave very similar to delayed after depolarizations. They're blocked by ryanodine, and also they're enhanced by pacing. As you pace more rapidly, this m-SCR gets larger in amplitude. Also, if you administer caffeine, the m-SCR becomes slightly larger in amplitude, but more importantly, it occurs much more rapidly. So what I try to do is I try to think, well, what's happening at the channel level? We know that with caffeine, you'll have a much higher open probability. You'll see more openings. And this translates, we think, into an m-SCR, which has a faster release of calcium. And again, you got to remember, we're measuring this not in one cell, from thousands of cells. All right, so these are the calcium substrates that we measure in our model. Here's a schematic of the rat heart. Here's an image of the mapping field where we're measuring action potentials, and also we can take pictures of the anatomy. The border zone surrounds the scar. The scar's here. This is non-infarcted tissue, not normal, non-infarcted. And I drew the border zone here surrounding the scar because sometimes that's what people associate with the border zone. But we've changed this a little bit. You actually can measure calcium transients and action potentials in the scar. And there's no reason to think why that couldn't be border zone as well. So we include that in our definition of border zone. So we see a greater incidence of spontaneous calcium release in the border zone. Here's a very nice example of that. You pace rapidly, you stop pacing, you get this spontaneous calcium release. Triggers a beat, spontaneous calcium release, triggers a beat. And here you have a spontaneous calcium release that was not big enough to trigger a beat. Over many animals, I think these summary data are from about 8 animals. There's a much larger spontaneous calcium release amplitude in the animals with ischemic cardiomyopathy compared to shams. The other substrate is alternans, as I mentioned before, and as you might expect, alternans is greater, calcium transient alternans is greater in the border zone compared to remote, that's further away from the infarct, and the remote is very similar to shams. Actually, it's true for the spontaneous calcium release as well. So you're more likely to get large SCRs in the border zone. You're more likely to get alternans in the border zone. Both of the border zone will be larger than both remote. The incidence of each will be larger in the remote in the border zone compared to the remote, which will also be larger than shams. We also see a higher incidence of triggered activity. So that's a real arrhythmia. And someone may argue, well, this isn't spontaneous calcium release, but it's not an arrhythmia. We actually see a greater incidence of triggered activity coming from the border zone in ischemic cardiomyopathy in this model compared to shams. So this is something that has been reported previously. So this isn't something new. But nevertheless, it's something that we did have to establish for our model. Well, we felt as though we had to see whether or not this was true in our model as well. For example, Peng Cheng's group has shown ectopy originating right near the border zone of MI. This is using optical mapping techniques. hint to careers has showed triggered propagated contractions from pectinate muscle in and around the infarct zone. Dr. Boyden has shown triggered activity in Purkinje cells taken near the infarct zone. And even here, Dr. Efferman's group has shown alternans that the original sites of alternans Ph.D. located in the infarct region and infarct adjacent regions. So it's been shown, nothing new here. We see something similar in our model as well. So the big question is, well, what's special about the border zone that's causing spontaneous calcium release and causing alternans as well? And this really is what we're interested in. What are the underlying molecular and cellular mechanisms of these substrates specific to the border zone? And our focus has been on oxidative stress. And I'll explain to you why. And a lot of these data are new data. Some have been published in abstract form, but also some of it is unpublished. So one of the first things we did was measure reactive oxygen species in our model. We used a fluorescent indicator of ROS. It's any kind of ROS, it doesn't matter. And what we found was an increase in ROS near the border zone of our hearts with MI. Now, in this technique, we used a method very similar to optical mapping, where we perfuse the fluorescent indicator to the whole heart. It's a Langendorf perfused heart. And someone may say, well, you have no ROS in the scar. Well, maybe that's because there's no dye getting to the scar, because this is a permanent ligation. So we also took tissue samples where we sliced the heart, and we used the same indicator, but applied the indicator uniformly to the tissue. And I hope you could see here, there's this area here where there's intense green fluorescence corresponding to really the whole scar of this heart. So ROS is elevated in the border zone. Again, this is something that has been shown before. We were interested in measuring ROS using optical mapping techniques so that we can then correlate this with the electrophysiology that we measure using really the same setup. In a collaborative study with Dr. Bjorky's group at OSU, we looked at alternans and their model of sudden death. It's a model. Dr. Billman has developed this model over many years. He infarcts these canines, he infarcts them, and then to make matters worse, he exercises them, and to make matters even worse, he inflates a balloon in one of his remaining patent arteries, and some will get VF and others will not. Okay? And that's how the animals are divided. This is an animal that never developed VF. Here's an animal that developed VF. And you can see here that the alternans is greater in the animal that developed VF compared to the one that didn't develop VF. Now, they also measured reactive oxygen species in cells isolated from the border zone and part of the remote zone as well. And you could see that ROS is elevated in cells taken from animals that have VF compared to those that don't. And below that, they're showing, this is a graph of ryanodine receptor free thiols. For now, think about it as a modification of the ryanodine receptor by the ROS. And a low number is bad. A low number of free thiols is bad. So if you, for example, use a strong oxidizing agent, you'll get a very small number of free thiols. Use a very strong reducing agent, you get a very large number of free thiols. These are sort of the upper limits, the ceiling and the floor of your response. And what you can see is that the ryanodine receptor is modified in a worse way in the VF animals. There's fewer free thiols, suggesting that this redox modification of the ryanodine receptor may be responsible for the alternans that was measured. So this was another clue that, for me, we did these studies some time ago. So this is published in 2009. We actually did the experiments, I think, 2 years earlier. And this started to get us thinking, me thinking in particular, that maybe I would be, this is something that I should look at, because I know alternans, and this may be important in models of MI. So what does ROS do to calcium regulation? That's a question that I thought, well, we should know about this. This should be known, but it's really not a lot known. I'm talking about the calcium regulatory proteins, not membrane proteins, calcium regulatory proteins. And this was a study where they looked at exogenous H2O2, which is a very strong reactive oxygen species. And they looked at calcium regulation. And they found that H2O2 reduces calcium transient amplitude. It increased diastolic cytoplasmic calcium. And also, importantly, reduced the rate of cytoplasmic calcium removal. Okay, so that's the decay phase of the calcium transient. It slows it down. And when I read this, I was thinking, oh, you know, we've looked at the decay phase of the calcium transient circuit expression and have related that to alternans. So it's a little bit of a clue there. Now, interestingly, they found when they controlled for SR content, so sarcoplasmic reticulum content of calcium, when they controlled for that, they found that H2O2 reduced spark frequency. So the open probability of the ryanodine receptor actually decreased in this example. This isn't true. This is not a uniform finding. And now I did throw in a membrane current, L-type calcium current. This is increased with H2O2. We know it, although it's not an SR regulatory protein, it does affect calcium. So we certainly need to consider it. So based on these data, I would expect that, in summary, there's a decrease in calcium removal, which they say leads to a decrease in calcium content. That's why you get smaller amplitudes, etc. So based on this, I figured, well, alternans, this could explain why alternans increases, but it doesn't explain why m-SCR activity would go. You would expect m-SCR activity to decrease, but we actually see an increase. So it may explain part of our results. So where does ROS come from? It doesn't come just from H2O2 that you sprinkle in your bed. There's many different sources, and I just want to focus on a few of these. Recently, it was a nice science paper where they showed that mechanical stimuli can activate NADPH oxidase, or otherwise known as NOx, and produce ROS in and around the ryanodine receptor and make the ryanodine receptor leaky. The increased open probability. So that's a little bit different than what I showed you before. Also, very recently, Yorkie's group showed that, interestingly, we know that digitalis is associated with spontaneous calcium release. But they suggest a totally different mechanism by which digitalis activates NOx and can make the ryanodine receptor leaky. So that was kind of interesting. Also, there's a very important enzyme, xanthine oxidase, which I'm going to be focusing on. There was a study by Gonzalez where they showed that xanthine oxidase, which is located in the SR. So this xanthine oxidase is producing reactive oxygen species in the SR. Reactive oxygen species, by definition, are highly reactive. So their sphere of influence, the xanthine oxidase's sphere of influence, is going to be localized to the sarcoplasmic reticulum. That's where all the calcium regulatory proteins are. Okay, so we're very interested in that. Also, H2O2 through the Fenton reaction will produce reactive oxygen species to these transient metals. So someone may think, oh, well, an antioxidant should work well then in preventing sudden death, but that was shown not to be true. This is a clinical trial, the HOPE trial, and they looked at what would be the effect of vitamin E, which is an antioxidant, on cardiovascular events. And surprisingly, they found that in patients at high risk for cardiovascular events, treatment with vitamin E for a mean of 4.5 years, is a long time, has no apparent effect on cardiovascular outcomes. So an antioxidant, surprisingly, isn't making matters any better. Okay, so this made us think that, well, maybe ROS maybe can explain alternans, can explain spontaneous calcium release as far as we can tell. So it must be something else going on in the border zone. Then we became interested in nitric oxide. Okay, nitric oxide is very important regulator of oxidative stress, just like ROS. What does it do? There's several different signaling pathways. You have the classic signaling pathway by which nitric oxide activates cyclic GMP, and you get where you'll reduce smooth muscle contraction. This is the classic mechanism by which nitrates work. You get a vasodilation. NO can also combine with superoxide and form peroxynitrite, which is a very powerful oxidizing agent. And then finally, nitric oxide can nitrosylate cysteine residues. And that's what I'm interested in. And we'll see why in a second. So nitrosylation is the addition of an NO group to a cysteine thiol side chain. It's a nice example of a post-translational modification of a protein. It's akin to phosphorylation, so it's very similar to that. It's reversible. You can have de-nitrosylation. At that point, you have a free thiol. Remember the plot that I showed you with the free thiols? When the free thiols was down, it was bad. Well, what can happen to these free thiols? Several different things. There can be glutathionation, which can occur. Disulfide bonds can form between other cysteine residues. And then, interestingly, you can get further oxidation by, for example, reactive oxygen species. So you can imagine, so what does this have to do with ROS? The way we like to think about it is that you have potentially either the protein is going to be nitrosylated or oxidized, one or the other, and there needs to be this balance between the two. So normally, there's a balance between NO and ROS. However, in disease, you get an imbalance. What happens is you have too little NO, proteins are denitrosylated, and if you have too much ROS as well, the proteins will be oxidized, and that will affect their function. Another bad thing about oxidation of cysteine thiols is that it's irreversible. As you can see here, S-nitrosylation is fairly reversible. Disulfide bonds are less reversible, and then finally oxidation is even less reversible. So once a protein is oxidized, it's going to be very difficult to reduce it in this way, when it's oxidized in this way. Whereas nitrosylation is fairly reversible. So oxidation is a bad thing. And one way to think about it, one way we like to think about it, is that the nitric oxide actually protects the cysteine residue from being oxidized. So you need that NO there. Very important. And this has been an idea, this is not our idea, this is an idea that's been proposed by several different groups. Okay, so I told you where the ROS is coming from. Where does the NO come from? Because you have to have this fine balance. Well, NO comes from nitric oxide synthases. There are 3 different isoforms. You have NOS, or NOS1. This is located primarily in the SR. It's located closely also to, this is xanthine oxidase, which I told you before was located in the SR. You also have eNOS, or NOS3, which is located primarily in the membrane of the cell. And then finally, there's an inducible NOS that really isn't only, that is absent and only becomes present when there's an inflammatory condition. So it's important in disease, and this has been shown by several different groups. What happens in, for example, myocardial infarction, NOS1 translocates to the membrane, and the inducible NOS, or NOS2, is upregulated. What's important here is that there's a decrease in NOS, hence a decrease in NO in the SR. Okay, so we already have a condition here in MI where you have less NO, potentially more free thiols that can be oxidized. Now here's an example of that. So here's a study by Gonzalez where they looked at the S-nitrosylated ryanodine receptor. So they looked at how much of the ryanodine receptor is nitrosylated. This was decreased in this. This is a different model. It's a spontaneously hypertensive heart failure rat model. So in this situation, the ryanodine receptor is less nitrosylated. Think about it as maybe being less protected from being oxidized. And we looked at nitrosylation of the ryanodine receptor in our model, and we also found that S-nitrosylation of the ryanodine receptor was reduced compared to sham. So in our model as well, the ryanodine receptor seems to be less protected. So here's where we thought about this and realized that MI is, as we know, is very complicated. We have many different substrates going on here, and we wanted to simplify our work. And based on a few other studies that were done as well, looking at the effect of decreased nitrosylation of the ryanodine receptor. For example, there's another study by Gonzalez and Josh Hare's group, where they looked at the nitrosylation of the ryanodine receptor in the knockout mice. So in a NOS1 knockout mouse, the ryanodine receptor S-nitrosylation is decreased. Now, interestingly, if you knock out NOS3, which is located at the membrane, okay, there's no effect on ryanodine receptor nitrosylation, which is what you would expect based on where the isoforms are. And they also found in this mouse, the NOS1 knockout mouse, they saw an increased incidence of both spontaneous calcium waves and also, here's a very nice example of, this is a normal animal, and this is a NOS1 knockout, where you get this spontaneous activity. And this was done in isolated cells, primarily. So what we decided, one of the things that we wanted, we were aware of this, and we said, well, let's see what would happen in a whole heart, rather than just isolate. Because I know, give me a single cell and I'll get 1 million DADs in it. But give me a whole heart and it's much more difficult. So let's see if this is relevant in the whole heart. So what we did is we did this study, not in rats, we did this in guinea pig hearts. And we looked at the de-nitrosylation and m-SCR activity in the whole heart by using this agent that specifically inhibits nNOS, or nNOS3, which is located in the SR. And this was using optical mapping techniques. In a control guinea pig heart, you pace rapidly. No matter how fast you pace, the best you can get is a spontaneous calcium release. That's it. No triggered beats. Even with a elevated amount of calcium. However, when you inhibit nNOS, you get very nice triggered beats that we think are due to calcium release. I'll show you an example why we think that's the case. And if you look at a lot of animals under similar conditions, it's only with SMTC, or only when you inhibit nNOS, that you get triggered arrhythmias. Okay, so normal heart, deplete or inhibit nNOS, you get arrhythmias. Specifically, you get spontaneous calcium release, arrhythmias. Okay, so this is for the optical mappers in the audience. We have a few questions about how do you know if a beat is triggered by spontaneous calcium release or not? And here was our approach to this problem. So here's the same example, I think, that I showed you before in the previous slide. Pace rapidly, you stop pacing. Here's a spontaneous ventricular beat, V1, V2, V3, and V4. That's the ECG. Below that are calcium recordings, calcium transient. Here's the spontaneous calcium release. Now it's a little obscured by the calcium transient. Next beat, you get this rise in diastolic calcium, then a beat. Here there's no rise, and then on this beat you also get another rise. We measured the slope of this rise of calcium that preceded the calcium transient. And interestingly, the slope was large and then decreased with time. Now if you remember early on when I showed you the spontaneous calcium release with or without caffeine, remember the slope of the m-SCR was greater with caffeine than without caffeine. Also, if you pace rapidly and stop pacing and you get oscillations of DADs, they get weaker over time, they dissipate. We think the slope is related to the strength of the spontaneous calcium release. So what we did is we then took these 4 beats, V1, V2, V3, and V4, and we mapped the activation time. Okay, so these are contour maps of activation time, which correspond to our mapping field. And the asterisk is where the impulse originated from. So here. Looks ectopic. Hard to tell for this beat. It may be coming off the field of view, we don't know. These are hearts where we've frozen the endocardium and mid-myocardium, so it can't be coming from deeper. Okay. So this is an ectopic beat that's originating within the mapping field, just like this one and just like this one. And then what we did is we started, we were measuring spontaneous calcium release from hundreds of sites. Let's map the slope of the spontaneous calcium release, because we think they're related to the strength of the spontaneous calcium release. And sure enough, when we did that, we saw a very close correspondence between the maximum slope of the spontaneous calcium release, the location of the maximum slope of the spontaneous calcium release, with the origin of the triggered beat for V1, very close correspondence for V2. V3, there was no correspondence. We think that this beat is coming from off the mapping field somewhere. We don't know where, we don't care. And finally, the last beat appears to be calcium mediated as well. So for those who have asked, that's sort of our approach, one way in which we sort of sort out, is this a reentrant beat? Is this a calcium mediated beat? So we believe that in this model, when we inhibit nNOS, we're getting these triggered beats that are due to spontaneous calcium release.
So do you also see the reentrant type of alternans, or because both things can be going on in your model, right?
You mean we see alternans?
Right.
Yeah.
So how would you distinguish between those? Because it looks like every single one of those beats is coming from the
Yeah, yeah. So let me go back and make sure I understand your question. So yeah, these beats are coming from different
So this
sites.
is triggered. You're
Yes.
arguing that it's
Yeah,
triggered.
yeah.
So if you were to see alternans, how would-
Alternans in like the calcium transient amplitude, for example, or
Right.
alternans
in the arrhythmia.
So you mean, for example, the arrhythmia, the site of origin of the arrhythmia alternating from one beat to the next?
Right, so you'd have calcium reentry, basically, alternans reentry in.
I don't know if I would call that calcium alternans and reentry. Remember, the connection between calcium alternans and reentry is that you have a calcium release alternans. It's not where it's starting from.
Mhm.
It's just the release is alternating in magnitude from one beat to the next.
Mhm.
And that affects APD, which sets up the gradient for unidirectional block and reentrant excitation. So I wouldn't expect the site of origin to alternate. Okay? And actually, if you look at this, I don't show you the beats during pacing, but I don't think they were alternating the amplitude. Any other questions? Okay, so we had to confirm that this SMTC actually was having an effect on the RyR receptor, and it was. There was a decrease in the S-nitrosylation of the RyR receptor with SMTC compared to control, so we know we were de-nitrosylating the RyR receptor. And also in isolated myocytes, we tested whether or not we would see after contractions, and with SMTC, you do see after contractions, after you pace, you stop pacing, you get these wonderful after contractions. And in an animal treated with SMTC plus GSNO. GSNO is an NO donor, okay, that's very stable. Typically, NO donors are very unstable. They just liberate NO right away. Very difficult to use. GSNO will liberate NO in a very stable fashion. Also, it'll liberate NO in the proximity of the SR. So here what we're doing is we're depleting NO from NOS, and we're adding an NO donor. Okay? And this decreased the after contractions, further suggesting that it's this de-nitrosylation that's the cause of these spontaneous calcium releases. What does it do to calcium transient alternans? Well, it increases calcium transient alternans a little bit, as you can see here. The effect isn't as dramatic as with spontaneous calcium release. All right, so if you remember before, I said there's this balance. So NO goes down, the balance seesaw goes up, and you have this ROS that piles on top and weighs down and shifts the balance. So, okay, I'm de-nitrosylating, but where's the ROS coming from? Especially in the animals that were treated with SMTC, right? Where's the ROS coming from in this case? Well, it turns out that if you deplete NOS1, NOS1 normally inhibits xanthine oxidase from producing superoxide, which is a reactive oxygen species. So when you deplete NOS, it takes the brakes off xanthine oxidase. Xanthine oxidase then can produce superoxide. Given its proximity to the RyR receptor, what we think is happening is that you are depleting NOS, de-nitrosylating the RyR receptor, and then you're also taking the brakes off xanthine oxidase, and it's going to start producing superoxide, which is then going to oxidize those free thiols. And to show this, we used another agent, allopurinol, and here's a control, pace rapidly. You can see here there's no alternans in there. Hard to tell, maybe we cut this one off, but there's no alternans in these last two beats. Control animal spontaneous calcium release. As I said before, that's about all you'll get. With SMTC, You get the triggered beats, spontaneous calcium release. SMTC plus allopurinol. Allopurinol is, it blocks xanthine oxidase. It's also a treatment for gout because the two are related. And we found then that, interesting, in many experiments, that the arrhythmias that were induced by SMTC were suppressed by SMTC when you add allopurinol, which is blocking, specifically blocking xanthine oxidase. So you're denitrosylating, but you're cutting off the source of ROS. And the arrhythmias go away. Now some of you may be thinking, oh, okay, I'm going to go out and file that patent because allopurinol looks like it's a winner. Unfortunately, oxypurinol, which is related, failed. Someone already tested this. Oxypurinol failed to demonstrate benefit in unselected patients with moderate to severe heart failure. So allopurinol, blocking of xanthine oxidase by itself isn't sufficient. So it's possible, one might imagine, that you need to not only decrease the ROS, but you may also, maybe there's other sources of ROS, and maybe you need to, in patients, right? This is patients with real disease. These aren't patients that are on SMTC, right? There's probably other sources of ROS, and maybe what you need to do is not only block xanthine oxidase, but also maybe protect the RyR receptor by using an NO donor. Maybe the two in combination will work. So finally, the last few experiments that I'm going to show you are some experiments where we added exogenous ROS to our preparations. Here's a control. Again, that's all you get, no more. With H2O2, interestingly, you'll get triggered beats, spontaneous calcium release looks a little bit bigger. The slope certainly is increased. And likewise with SMT alone, you get the triggered beats and spontaneous calcium release. And where we noticed the greatest difference in all of these experiments was when we combined the two, the exogenous ROS with the denitrosylating effect of the SMTC. Here's a wonderful example of, here's the last pace beat, and you get this, if this is all driven by calcium release, this is a pretty bad arrhythmia for that. It's very hard sometimes to explain how spontaneous calcium release can sustain arrhythmia. And we think in this case that we're getting a very nice sustained arrhythmia. Look at all of these spontaneous calcium releases that are preceding the calcium transient. They're all over the place. Okay? Very dangerous situation. And of course we saw this in several different experiments. These are just summary data. And finally, we also saw a big jump in alternans. We combined the two. SMTC by itself, as I showed you before, increased alternans. H2O2 also increased alternans a little bit. But when you combine the two, you get this very large increase in alternans, not only at similar cycling, but you get a decrease in the threshold. It's easier to get alternans when you combine the two. So that's where we stand right now in terms of trying to resolve this puzzle. We have to come back now to MI and try to sort out what's going on with the redox modification, not only the ryanodine receptor, but also some other calcium regulatory proteins. What are some other possibilities? Certainly CAM kinase. You can't forget about CAM kinase. CAM kinase is activated in MI. CAM kinase will affect many calcium regulatory proteins, but also CAM kinase can be activated by oxidation, shown here, with the addition of H2O2. It can be oxidized and maintain its activity even in the absence of calcium. So it's a way that CAM kinase can maintain its activity without calcium. And when we looked at nitrosylation of important proteins, we found out that, well, there's a lot of ion channels that are regulated by nitrosylation, and you can see that the effect is quite varied. There's a decrease in the peak sodium current with nitrosylation. The late current is increased with de-nitrosylation. All of these currents are affected by nitric oxide and potentially nitrosylation. So in summary, abnormal calcium regulation may be a very important arrhythmia substrate associated with chronic MI, in addition to the substrates that we already know about. Also, we think that elevated ROS, along with de-nitrosylation, may explain in part these calcium-mediated substrates. So this sort of suggests that this Nitroso-redox balance that we call it may be a very important and novel therapeutic target for the treatment of patients with chronic MI. There's a collaborator that I have that a case where we're developing new therapeutic strategies based on these data, so it's very interesting. But we really have a lot more to learn, and additional studies are going to be required to determine the mechanistic relationship between oxidative stress and ventricular arrhythmias. And here's people that I have to thank. Michael Cutler is a trainee. He's now an electrophysiology fellow. He's the one who really put together a lot of these studies in his mind. And he thought that, well, we should look at this de-nitrosylation in whole hearts, which hadn't been done. And then Xiaoping Wan, who's in my lab, and Plummer, and also Lance Wilson, who does a lot of the alternans work. And finally, Jonathan Stanlow over at University Hospitals, who's really he discovered nitrosylation and is world famous for that area, and he's been a really outstanding resource. And that's it. Thank you very much.
Thank you very much. It's really an interesting and you know, another angle looking at the infrared.
Just what we need, another angle.
It's interesting. Maybe not the last word and not the last angle. There'll be more. So these things are very complex. You know, if you think about this whole thing has neural effects that are changing with the infarct, which is not taken into account. There's a lot of things going on. So, but it's very interesting. Yeah, Catherine.
Would you expect then to find nitrosylation of the ryanodine receptor or other calcium handling proteins in the infarct border zone if you measured by mass spec, for example? Or since you mentioned that nitrosylation is reversible, that it would be something that, you know, would be difficult to measure if you actually took some human tissue, for example?
Yeah, I don't think it's, I don't know the details of the assay, but my colleagues this is their bread and butter, measuring nitrosylation of proteins, and the mass spec technique, there is something published using mass spec, and there are proteins that are nitrosylated. That's been shown. And my collaborators at CASE, they have several different techniques for measuring. So I feel comfortable that if you were to measure it in human tissue, you would see that there is, in diseases, a decrease in nitrosylation. So I feel pretty comfortable with the techniques.
And would you expect to see it also in heart failure then?
Yes, yeah. A lot of people don't distinguish, especially with the ischemic cardiomyopathy, it's really the same thing. So it's been shown in both. In animal models, it's been shown. Yeah.
Very nice.
Thanks.
What do you think is the connection between the spontaneous calcium release and the DADs?
In what way?
Like what membrane currents would be responsible for this translation?
Well, I didn't measure that, but my guess it would just be spontaneous calcium release, and sodium calcium exchanger just does its job. It removes calcium because that's what it's been told to do. That's what it's built for, and it happens to generate a current in the process of doing that.
So that would be the only current, or it would be?
Would there be others?
Right.
Maybe.
So at the molecular level, do you have any ideas, or anybody has any ideas why the ryanodine receptor becomes more leaky?
No, that's what when we were talking with your molecular dynamics group. what happens, how the protein changes its conformation when it's nitrosylated. Also, I didn't mention it here, but hypernitrosylation, some people also believe is bad. So how does the protein change when it's hypernitrosylated? When it's oxidized, how does the protein change? I think we know a lot more in the skeletal isoform, they know a lot more, but it's different in the cardiac
It'd be interesting
isoform.
to try and see what causes the leakiness of this random receptor.
Yeah.
From the structure perspective.
Yeah, it's very interesting. Yeah,
Are thiol groups abundant in the ryanodine receptor?
Yes, there's a number of them. And part of the problem is we don't know which ones are important. There seems to be some that are important and others that are less important.
And do you expect that this nitrosylation, are these, so nitric oxide can cross a membrane pretty easily, right?
Yeah.
So you don't expect that it will be limited to the surface facing inward to the SR?
Well, it depends on where the NO is coming from, right? If it's coming from nNOS, which is inside, right? So it's gonna react with the cysteine residues on that side of the protein, where the inducible NOS and maybe the NOS3, which is outside, could affect it as well. So the reactivity is the sphere of influence, I like to think, is going to be very small. So it's going to be highly dependent on where the NO is coming from.
So presumably it's just the thiol groups that are on that face.
I would imagine, yes. So one possibility might be to think, well, if, for example, I decrease nNOS inside, and something about those cysteine residues on the inside, yeah, could be. Yeah. Yes.
So if I understand your talk, this borderline area plays an important role in this process, or it has some activities that are unique to this area?
The border zone of the
Border zone.
MI, yes.
Now, have you looked at that, or does it even make sense to look at that over time? Do those things change? Because you're looking at chronic MI.
Yeah.
And would that make a difference?
Yeah. I think it may be different for acute ischemia versus 3 to 5 day versus maybe a month or 2 months. Beyond that, it could be. It's a very good question. I think really we know, despite the fact that MI is the big killer, we really know very little about the substrates associated with it and how it changes over time. I'm sure ECGI would be.
Oh, we know structural remodeling is a good detail. And also electrical remodeling of the properties of ion channel at different times after the infarct. There's a lot known about the 6-day-old infarct.
Yeah, mostly that.
Yeah.
But
But even the chronic infarct, people have done most of the ion channels that go through changes in remodeling of the 6-day-old. They then recover and they become normal.
Yeah.
And there's sort of a balance shift or crossover between electrical remodeling of ion channels and structural remodeling of the infarct due to scars and gap junction uncoupling and all of that. So they intersect somehow around 6 days to 10 days to 2 weeks.
Yeah. And also, it's not just the border zone in the scar that's got something wrong with it. The remote zone also has something. You get hypertrophy in the remote zone.
Cannot be normal either because it experiences different stretch than normal. But in the border zone, it's the whole name. The border zone thing is the names. I think I think the whole scar has all kinds of different regions in it, so you can't. It's not so well delineated as just around the scar. We see with ECGI we see a lot of fractionation coming from the scar.
Yeah. Well, that's been our experience. I told a few of you that one of our surprises was that you could measure optically action potentials from the scar. You look at the tissue and you swear you would never be able to measure an action potential from this tissue, but you measure an action potential.
Right. And clinically, people find fractionation inside the scar, deep scar, the ablution. And it's consistent with the model from Jacques de Backer.
Yeah.
It has all those.
It's more like border zone.
The border zone is not so well defined. I think this was a mistake to put some emphasis
Yeah, anatomically, it's on the border of the scar.
It's not so well. It's not a good. It's not a good tool. But I think this is an added complexity. And then if you add to this also hormonal changes and because the scar now has very different innervation than what it had before. All of the cancers.
It's
It's a
complicated.
complicated story. Anyway.
Is it possible that myofibroblasts can contribute either calcium or ROS?
That's an interesting question. Do you know? I don't know. I know that people are attributing a lot of abnormalities to these myofibroblasts. They're not just there and blocking impulses. They're very active. They could be contributing ROS. That's a very good point. It could very well be.
It's possible.
Yeah.
So following up Dr. Rudy's question,
Yeah.
in the mapping studies, how did you define what caused the border zone and what wasn't the border zone?
Well, we started using the approach where we just looked at the edge of the 2 millimeters within the edge of the scar. But then we realized this is nuts. We're measuring action potentials from over the scar. So now we sort of use our new definition now is any place where I can measure an action potential or a calcium transient is border zone. If I don't measure anything, flat line, then it's scar, we don't care about it. But usually we can measure either an action potential or a calcium transient in the scar area. So we call it all border zone. Now the amplitudes will be much less because there's much less viable tissue there. The optical dyes don't perfuse as well there. But otherwise, it looks like there's some viable cells there. So our new definition is wherever you can measure a signal, that's border zone.
And these border zone changes, how far do they extend into the remote myocardium? Is there gradient changes from the scar?
No, it's pretty delineated. Yeah. I'm sure if you looked at it with high resolution, you would see something different. But on the surface with your eye, it's pretty well delineated.
I think clinically it's part of, well, you know better than I do because you do, but clinically it's part of the reason that now people are not so enamored anymore in circumventing